maybe another minute or so.
Okay, sure.
Is the right, I'm trying to say, what screen is visible to you guys? The correct one or?
The
Seems
thank you
like.
sponsors.
Yeah.
It's
Sorry,
yeah. Oh yeah, that was better. That was better before.
Are we good now?
No, before was better.
Oh,
Yeah,
okay.
you have to swap. Yeah.
Swap it back. Okay.
There you go.
So welcome, everyone. I'm Tony Latai, president of the SFPM, and welcome to March 2023, our monthly seminar series, shortly presented by Olivier Elemento from Cornell. But first, just a few announcements. This is a little redundant from last month, but these are very important, so I hope you'll all pay attention. First, I'd like to thank the sponsors who make all of this possible. We are run as a very lean organization, and none of the officers or board members get any compensation, but we do need some resources to get some things done. And we appreciate the companies listed here for giving us support that we really badly need. This is a good time to remind everyone on board that you should that almost certainly you have not yet renewed your membership for 2023. So please go to the sfpm.io website and renew your membership. Along those lines, you can use this convenient QR code down in the lower right-hand corner to go directly to sfpm.io. But I also want to let you know at AACR down in Orlando, we will be hosting a reception on Monday evening. And it's just a really short walk from the convention center where there'll be anyway. There's going to be free food, drink, and opportunity to network with all your other functional precision medicine cronies, meet the board. It's a great opportunity to share ideas for what the board or the organization could be doing for you particularly. We exist to help you do your work better, get more attention, and to sort of raise the entire field. So it would do us a lot of good if you can go and save your spot here so we can have some idea of numbers. So if you just go to our website, sfpm.io, and sign up, we will know that you're coming and we'll save a spot for you. If we do get overbooked, we will have to prioritize letting in people who have saved a spot in advance. Finally, there is an important major scientific session on functional precision medicine at the ACR meeting, and you can put it in your calendar if you follow the same link to sfpm.io. Aaron has very kindly designed something you can put it right into your calendar where you can see Pam Becker, Alana Wellman, and Philip Stauber speak. I just want to encourage everyone on this call not only to come, but to encourage your friends or the people in your lab to come. It really is important. They keep track of who attends these sessions. And the bigger numbers we can bring to these sessions, the more likely it is we can continue the momentum and continue to have sessions at these major scientific meetings. So tell a friend and tell your friends to tell a friend to come to this session, Tuesday, April 18. New information is we actually have a location, Tangerine Ballroom 2 in the Convention Center. So, and as always, feel free to contact me or Aaron or any of the other board members if you have ideas. But for now, I'll turn it over to board member Alice Sarania, who will introduce today's speaker.
And maybe I'll just add, Tony, to your comments that if anyone on the call is presenting at the AACR meeting, anything related to precision medicine, just contact us so we can advertise it also through our channels, through Twitter, etc. I think that would be great. All right.
Thank you, Alice. That's a great idea. Everyone, let us publicize you.
Yeah, we would love to do that. And so without further ado, please let me introduce Dr. Elemento, who is a professor of physiology and biophysics at Weill Cornell Medicine and Cornell University. Since 2017, he has been directing the Englander Institute for Precision Medicine, which is this terrific multidisciplinary institute that brings together over 100 faculty members, spanning all sorts of basics and clinical departments at Cornell. Their mission is to use genomics, artificial intelligence, and patient-derived models to develop highly personalized medicine solutions for patients at Weill Cornell and affiliated hospitals. And so Dr. Elemento has been very successfully funded throughout his career. He has published over 360 papers, and I think he has this very unique expertise, really spanning all the way from the genomics to the kind of functional precision medicine element. And so we are very much looking forward to his talk today. Olivier, if you want to share your screen, hopefully everything
Yeah,
works
that sounds
smoothly.
great. I'll do that. Well, thank you so much for having me. Real pleasure to be here as always. And like you said, Alice, I'm going to try to give you an overview of our program. When we think of functional precision medicine as critical to everything that we do, the idea of really establishing function for for example, elements that we see in the genome when we sequence cancer genomes, as well as obviously directly assessing sort of phenotypes in response to perturbations. I think for us, we think of this as a kind of multiple sort of set of technologies and techniques that we want to use to personalize medicine. And so what I want to do today is to kind of give you a tour of these different technologies. Think of it as kind of a crescendo where, we start from genomics, which is maybe more traditional, but, the goal is really to complement genomic sort of techniques with the ability to test hypotheses using patient-derived models, which I think is very complementary, as you will see. So we're not, using one way to try to personalize medicine. We're trying to kind of use complementary different solutions. Hence the content of a talk, which is going to start with an overview of how we sort of think of precision medicine, let's say now, and I think how it's done in multiple centers, which is, a lot of it is really focused on using DNA sequencing. We're trying to push the envelope there, but obviously are seeing a lot of limitations to a just DNA type of approach. And so that's why we're investing in additional technologies. One of them is the ability to image tissue at single cell resolution to assess the complexity of tumors at single cell resolution, including the discovery of the different types of non-tumor cells that you have within tumors. And as you'll see, I think they tell us a lot about, for example, response to chemotherapy, response to therapy. But I think that we also are sort of starting to understand that we need to be a bit humble, is that we can measure things kind of statically in some ways, but it doesn't really tell you the whole picture. I think the ability to really perform almost mini experiments, almost mini clinical trials on patient-derived models to assess what a patient is going to respond to, I think is critical, because there's so much that we don't understand about, the physiology of tumor. So just measuring and predicting, I think is hard, but it's kind of perturbation approach that, has become very, very popular. I think complements the other approaches I'm going to tell you about really nicely. So our mission is really to almost try to reinvent medicine to make it more personalized. Really, I think we can really think of the future of medicine as much more personalized, where we can assess the disease with each individual patient and treat patients based on that understanding of a disease and not based on the kind of average understanding of lots and lots of patients with the same disease. I think this is really the way to go in the future, and I think we'll be looking, sort of in, let's say 25, 50 years from now, we'll be looking at what we're doing now in terms of treatment of patients, maybe with a obviously, reflecting that it was not probably the best approach. I mean, it's obviously, it makes a lot of sense to kind of start in this way, but I think now, we have lots of ways to really improve how we explore the disease within each individual. And I don't really need to sell this too much to this crowd, right? Cancer is extremely heterogeneous between patients. That's just an example here on this slide of two patients with breast cancer. They have about the same age, and basically according to kind of things like morphology and where the tumor is located, they have the same disease, right? But in fact, if you look at the set of mutations that each of those patients has, and this is just showing the top of a mutation list of mutations, those patients have completely different mutations, like no overlap, not a single mutation or overlap, not even actually a single gene overlaps between those two patients in terms of where the mutations are. So, quite clearly they have a different disease, right? And therefore, I think we need to treat those two patients in very different ways. Thankfully, now we can really assess disease within each patient with a tremendous amount of detail using high throughput sequencing, using spatial kind of techniques like spatial proteomics, and have access to some really nice technical tools to analyze the data that comes out of these technologies. For example, some really modern AI tools are good at integrating, let's say, data from different modalities and making predictions. And these are the kind of tools that we use over and over again. In addition to this, the technology to make patient-derived models has also improved. There's high throughput robotics, that we can use, and I'll show you essentially a little bit of all of this during my talk. So precision medicine, at Cornell, the way we implemented it, I think we have actually a substantial amount of control when it comes to, access to tissue and biopsy and surgical samples. We have a great biobank that we work with for immediate access to fresh tissue. And so that's really been, quite instrumental to the success of us, the more functional aspect of, what I'm going to tell you about towards the second half of the talk. And obviously we do multi-omic, let's say multi-modal sort of type of analysis with the idea of combining all this information eventually into a report, which still needs, discussion among experts. Not quite something that is, kind of ready to provide to physicians directly, but needs, discussion because of the complexity of the data, but eventually is giving rise to highly personalized treatment recommendations. We enroll patients into research protocols just to make sure that we can reuse all of this data here, published papers and so on. In fact, I'm showing 2,000 patients enrolled now. And it's actually closer to 4,000 at this stage. So it's a very large cohort that we've enrolled into our precision medicine protocol. So as I say, the bulk of the analysis that we've done in the past, if you want, we're focused on DNA sequencing of the tumors. We've actually been among the first to implement whole exome sequencing as a clinical tool, here at Cornell. We've learned a lot in the process of doing this. One thing that we learned is that genomes are very complex, especially advanced cancer genomes, very, very mutated. Like you see in this particular case here, that's a prostate cancer genomes and advanced prostate cancer genomes with looking at copy number alterations. And as you can see, there's a gigantic number of mutations in those genomes. And it is a real challenge to kind of narrow down all these mutations to the mutations that are important and can be targeted. And so we do this mostly by essentially looking up those mutations into in a database. There's actually not much AI involved, in doing this yet, maybe in the future, but not right now. But that information goes into a personalized genomic report, which catalogs mutations based on what the database tells us. Important mutations at the top, less important mutations in the middle, and then the non-important, presumably non-important mutations at the end. And we've issued several thousands of those reports in the past. We've actually, this is kind of more playing, but we've actually been playing with tools like ChatGPT to kind of assess whether, there's ways to maybe, get an automatic readout of genomes based on mutations. And I think there's very limited success in doing that, pretty kind of underwhelming ability to kind of, use ChatGPT. And that's just an example, but I think, I believe in technologies of this kind, but I think they need to be trained better on, actual patient data and patient response data. One thing that we've realized in that kind of, starts going into a functional aspect is that the DNA doesn't tell you the whole story. And that's something that I'm going to keep repeating over during the talk. One thing that, we realize is important to assess is the extent to which mutations are functional, if you want, or are driving mutations. And one way to assess this is by using gene expression profiling of the same tumor. This is a busy slide here, but it shows you a transcriptomic report for a patient that has an ERB2 gene fusion here. We wanted to know whether that gene fusion was active. Thankfully, we had actually a set of genes that we know are downstream of ERB2 through manipulations of ERB2 levels, overexpression, for example. And so one thing that we can do is to ask whether in this particular patient, compared to patients that don't have an ERB2 mutation, do we see upregulation of all of these ERB2 downstream genes together as a whole, right? And we can use GSE as a way to do this. And we do see that, in this patient, and we do see that those genes, you see here at the bottom, tend to be upregulated as a whole, which basically I think for us is a very good functional assessment, if you want, that ERB2 is active within this particular patient, and therefore that targeting ERB2 may be a good idea. And I think this is, one example by which we can get, more to our sort of functional mutations, because just looking at DNA mutations is really, I think, very limited. I mean, especially in the context of metastatic tumors, which change locations during the course of a disease. What used to be a driver mutation may really not be a driver mutation once, the tumor has gone to some other location in the body. And we see that over and over again. I think prostate cancer is a good example. You have AR mutations that are driving localized disease, but, very unlikely to, for example, drive prostate cancer metastasis to the brain. There's no androgen in the brain, and so therefore AR mutations may be there because of what's sort of kept through tumor evolution, but they are not driving the tumor in the brain. So just looking at DNA is limiting in that sense. And we've established some nice techniques to be able to assess RNA expression, for example, within FFP tumors, which is still the standard in terms of the kind of tissue that we're getting. But I think we also realized, process of doing this, and I'll show you some evidence for this, that whole genome sequencing is really the future of this kind of DNA assessment. Whole exome sequencing, I think, is limited because you're basically capturing DNA, and there's some issues with the process of capture of DNA and inefficiency in some exons, for example. But also, I think there's a growing number of mutations outside of the genes themselves. I think something that I would call the dark matter of a cancer genome, which is a lot of structural variants that are important. A good example was this paper here that we published a couple of years ago now, led by Marcin Bielinski in our institute. It was an analysis of about 100 lung tumors that lacked driver mutations in the RTK RAS RAF pathway. So we realized that by doing whole genome sequencing of these tumors, whole exome sequencing wasn't able to find mutations in those cases. By doing whole exome sequencing, we see two things. One is that it turns out there are actually quite a few missed mutations by whole exome sequencing. These are examples here in exons that don't capture very well. The capture efficiency is very low according to whole exome sequencing. But I think the more important sort of finding was that there are actually a very large number of cases among those 100 cases with structural variants, including some affecting, for example, EGFR, which is a classical lung cancer gene, but in a way that essentially that was not really detectable using whole exome sequencing. Kind of very complex structural rearrangement like you see here, involving kind of nested the duplications and sort of mini translocations, and that led to essentially a very difficult mutational sort of assessment. Same here, you have for example, loss of certain short exons that was also extremely difficult to assess using whole exome sequencing. So we saw a lot of basically I think about 2/3 of those cases actually turned out to have a important driver mutation uncoverable by whole genome sequencing. And to us, that convinced us really that was the way to go. And now since then, we're deploying, I think, a lot of effort and energy and investments and resources to deploy whole genome sequencing in the clinical setting. This is just something that we started a couple of years ago, and we have a lot of data now, and we're actually going to start showing some of that data at AACR. But this is something that I'm not going to go into details today because of the fact that it's still a bit early. I want to really highlight the fact that again, DNA does not tell you the whole story. DNA is basically looking at mutations that are only in the tumor cells. But we all know here that a tumor consists of many cells that are not tumor cells, and that's a kind of very simplified example of what a tumor looks like. I think there is obviously, a lot of potential in assessing those non-tumor cells, the content of tumors in that sense, and understanding maybe whether, certain tumors may respond to, let's say, immune therapies, or maybe respond better to chemotherapies, once we understand the microenvironment of these tumors. The way we do this in our group is to use single-cell proteomics, which is the technology in the middle here, but we do also single-cell transcriptomics. The technologies are not as mature, in my view, as single-cell proteomics, which is why we use that technology. We're using the imaging mass cytometry technique, which is essentially using antibodies tagged with metals, metal tags, essentially, for each of the antibodies. We stain tissue sections with up to 40 antibodies at the same time. I'll show you examples of antibodies that we use. A laser in the instrument is vaporizing essentially the surface of a tissue, and the ionized metals go into the cytor for quantification of the amount of metals at different locations across the slide. And so that gives you a sense of the abundance of the markers that the antibodies recognize. And it gives you essentially abundance maps across the tissue that you look at. This is the instrument here. This is what it looks like. We have two of these. And this is the kind of tissue that we look at. You're trying to do this in reasonably high throughput format. So we're using this kind of tissue microarrays sort of format here on the right side, with each dot, each spot corresponds to one patient, and we can put multiple patients on the same spot. We typically put multiple spots for the same patient. We're doing this now, and this is really just one project, but we're doing this across many different types of tumors, in fact, also non-tumor tissue. And the results, I think, are pretty spectacular. I'm just going to show you one sort of short example in B-cell lymphoma, which is a disease that we know well, and that we've worked on for some time, that is also characterized by the fact that all patients get the same treatment. So, it's maybe easier in terms of sort of relating biomarkers, things that we can measure to response to chemotherapy. It's a combination of 5 different chemotherapy agents. We've been analyzing a large data set, about 400 patients with this kind of aggressive lymphoma. We have a lot of data on those cases, including mutations, RNA-seq, and clinical data. These are the markers that we looked at. So as you can see, they are, essentially markers that define, sort of help us define what the immune content of these tumors look like. But also, I think some functional markers, right? Some functional markers that tell you something about the functional status of cells, whether the cells proliferate, each cell proliferates, whether it's has a particular metabolic activity, whether it's expressing checkpoints, whether it's dying. So we can assess all kinds of functional statuses of individual cells using those functional markers. And then we have structural markers that tell us something about where you have blood vessels, for example, in other sort of matrix sort of type of involvement. This is what the data looks like. This is for normal tissue. That's a normal lymph node here. This is showing 4 markers at the same time, but just to show you that, we're using those 4 markers. This is, a subset of all the markers. There's 40 markers, but I'm showing only 4. You can see the cells. You can see that this is, for example, you can see macrophages, CD11C cells. The sort of the hole at the center of that's the nucleus. I'm not showing the DNA channel here, so you're not seeing the DNA, but these cells are not proliferating. They're not blue because blue is Ki67, but they're nicely sort of positive for CD11C. And you have some PD-1 expressing cells, some proliferating cells, and the green cells are the T cells. So pretty nice visualization of what's happening within tissue. And I love to see these. I love to see these maps. I love it because I think it really shows you what the disease. I mean, this is not disease here, but this is what the tissue looks like. I kind of think of this in the context of cancer as almost like a battlefield. Where you can actually see individual cells almost in the process of fighting each other. And I just love it because I think it gives us so many hypotheses, so many ideas about what's happening and how we can perturb maybe some of these interactions between cells. And maybe moving forward to maybe achieve better treatment. This is what tumor tissue looks like. I show you. These are the cores I was showing you before. And so this is only a small set of markers at the same time, but you can see some structure. You can see blood vessels. CD31 positive blood vessels here. The blood vessels is cut through the vessel. That's why you see this elongated shape. And so the analysis of this kind of data is really computer vision. It's actually, completely like non-sequencing type of analysis. We're doing a lot of analysis and sequencing data. That's like completely different type of skills required. We have to find the cells using segmentation. We have to essentially quantify the amount of signal within the cells or in the nucleus at the membrane, and so on. Once you do this, what you can do is to essentially establish a major cell type for each of the cell based on the key markers that they express. And for example, if you see that cells express CD20, that's it's a B cell. There are some markers that we chose that are very specific towards some immune cell types. This is a B cell lymphoma, so essentially most of the CD20 expressing cells are lymphoma cells. But what you can do is to go deeper, right? You can essentially stratify cells into different populations based on the combination of markers that they express. When we do this, we see a lot of depth, a lot of let's say granularity in the cell populations. We see, let's say, for example, 6 types of B cells in this B cell lymphoma. We see about, let's say, I don't know, maybe 11, 12 different types of myeloid cells, 5 types of stromal cells, and about 7 types of T cells. And it could actually go deeper and deeper in terms of subpopulations, but this is kind of at a reasonably high level. The kind of subpopulations that you see. And again, each row here is a specific population that is defined by the combination of markers that they express. So some of myeloid cells express, myeloid markers, plus maybe additional markers like VISTA, right, which is a checkpoint. So now we can go from the maps on the left side, which is just raw data, to the maps on the right side, which is essentially, where each cell has a specific identity, like we understand what each cell does. Obviously then the question is, what does that tell us? What does kind of a composition of a tumor tell us about the tumor itself? One thing that we can do is to classify or cluster tumors, not so much focusing on the tumor cells, in fact, ignoring the tumor cells, but completely focusing on the microenvironment. We actually didn't really have too many expectations when it comes to this kind of analysis, but when we actually do the clustering based on the composition of the non-tumor cells, we actually see 5 different types of microenvironment in this disease, which is, again, a surprise to some extent. We see a lot of cold tumors, tumors that don't have any immune cells, but we see essentially a lot of immune-rich subtypes, or let's say tumors, for example, CD4 T-cell rich type of tumors, tumors that have a microenvironment that's more inflammatory, if you want, and an interesting subset of lymphomas that have a highly cytotoxic type of microenvironment, which is essentially characterized by a lot of CD8 T cells, CD4 T cells expressing granzyme B, that's why we call it cytotoxic, but also a lot of immunosuppressive myeloid cells, and in fact also a lot of exhausted T cells. And you can convince yourself that this kind of microenvironments, are real and really kind of are as stereotypical as I showed you in the previous map by looking at examples. And you see this cytotoxic cluster here with a lot of these myeloid cells in the right side, core cluster with essentially very few immune cells. So we think it's real. I think another way kind of to convince ourselves that these clusters, these subtypes of microenvironments are real is that they correlate very nicely with survival. If you look at progression-free survival on the right side, you see that there are 2 clusters, 2 subtypes of microenvironment that correlate very nicely with survival. One of them is a cytotoxic cluster. This cluster that has a lot of immune, but dysfunctional immune cells. Actually, the patients who have this microenvironment do very poorly upon chemotherapy. What's interesting is that the second worst in terms of response to chemotherapy or time to progression is actually a very distinct type of microenvironment. It's this cold microenvironment that you see in green here. And so I think that to us, that was a bit of a surprise that the microenvironment was just so connected to survival. I think it really tells us that there is essentially some kind of synergy between chemotherapy and the microenvironment. In fact, based on these observations, we think that chemotherapy probably needs some kind of semi-functional microenvironment to be as effective as possible. If you don't have the right microenvironment, which I think is more like a normal microenvironment in some ways, you basically really limits your ability of chemotherapy to help patients. And I think that's an important lesson in our view, which also gives us some hypothesis about how to maybe reestablish a more functional microenvironment so that we can then sensitize some of these tumors to chemotherapy. And we're testing some of these ideas now. We can actually combine all these different markers with clinical data to assess the best way to assess survival, and we see that really this combination, even though the clinical markers are important, like age, still critical in terms of predicting survival, but we see very important markers in the microenvironment and also in the tumor cells that together actually give rise to a very, very accurate predictor of response to chemotherapy. And so that's also really, really important. We're trying to translate this kind of combination of predictors to a clinical assay now. Because, obviously, if we find a patient in the kind of in a non-responding sort of status, it doesn't make any sense to treat that patient with chemotherapy. Probably should enroll that patient into a clinical trial, more likely to hopefully respond to something and to help the patient. Another aspect of this kind of technique, which we are starting to explore now, is looking at compartments. Because, we see, things like this, here, look on the right side. If you could think of this tumor as like a supercells, this kind of cells are distributed almost like randomly, but it's not the case, right? If you look at this picture here, you see that on the top right, there's a very strong B-cell rich compartment, and the bottom left, you have a much more T-cell rich compartment. So there are actually compartmental structures in these tumors, and I think we also need to understand. We've actually just recently published a paper trying to get to that problem, to essentially be able to segment tumors into different compartments of cells that essentially have a similar composition. I'm not going to go into details about that technique because of time, but it was actually quite sort of helpful. We calibrated this technique in normal tissue where we had manual annotation. This was lung tissue here. We had a manual annotation of different compartments in the lungs, and the this technique really was able to recover this manual annotation really well. Actually, in tumors, when we apply this to, let's say, for example, a bladder cancer data set of images that we had in-house, it also really revealed several compartments that, likewise, we had manually annotated through our pathology department. And so it works well also in tumors. And we applied this to a breast cancer data set from an imaging single-cell breast cancer data set from the Boden-Miller group. And we're able to find actually quite a bit of structure in that data set, including, for example, some tumors with stromal compartments with a lot of T cells versus stromal compartments with very few T cells. And we see, likewise, a very good correlation with survival in terms of the abundance of those compartments within tumors. So I think it tells us that there is some biology, behind these compartments. I think we need to understand better. But I think that was kind of the first step, and now we're applying this to many more cases. Which obviously, again, this is a very orthogonal to the DNA analysis. But likewise, again, I think we always reminded that we can measure lots of things, but it doesn't mean that we understand all these things. And so I think there is a major need for a more sort of let's say functional approach to precision medicine that complements what I showed you before. And that's where our patient avatar sort of type of approach comes in. We've been actually working in this for some time, and now we're getting to the point where this is really being returned to patients, at least in a research protocol. But the idea has always been that we wanted to know whether we could potentially test thousands of compounds and drug combinations on the patient's tumor cells and assess what may be the most effective therapy for this one patient. This is really the ability to test many hypotheses. I always say, in patients, you can test one hypothesis at a time, and it takes a very long time to get the answer to your question, one drug at a time, or let's say one combination at a time. You can't, give patients too many rounds of different therapies. It's obviously, not practical by any means. So we need a better approach for doing this. And so a few years ago, we started this kind of systematic approach to trying to grow tumor organoids. I don't know if tumor organoids is really the best label, for this, but they would call them tumoroids if you want. There's different ways to depict them or to describe them. But we do this sort of side by side with sequencing, and in some cases, with patient-derived xenograft. But we do this systematically, and I think that's what makes us reasonably unique, is that we're doing this across lots and lots of different tumor types. These are just examples of 3D sort of cultures here. This is, we're trying to make these cultures in matrigel, like many of you, just as a way to preserve, the context between cells, which I think is really critical. As I say, what makes this effort reasonably unique is that we're kind of cancer agnostic almost. We're doing this across almost every cancer type that we can get our hands on. This is not really showing brain tumors because we have a colleague here, his name is Howard Fine at Cornell, who's focusing on brain tumors, and so we're working with him on brain tumors, but we're doing pretty much everything. And so you see this pie chart here has a lot of diversity in terms of tumor types. We've actually done now, but we have actually been able to successfully grow an organoid or tumor organoid for about 250 or so patients. And we're continuing this work and doing much more now. There's, I don't need, likewise, I think many of you have seen this kind of slide here, but there's tremendous diversity when it comes to the shape that these 3D cultured cells have. Within the same tumor type, there's actually quite a bit of diversity, and so these are just examples. We're doing this in, for example, a metastatic melanoma, and this is a slide that I like to show because it shows you, I think, the extent to which it's important to do these cultures in 3D format in matrigel. We actually compared 2D cultures versus 3D cultures. In metastatic melanoma, we see clearly that the contact between the cells is helping cells preserve the pigmentation of the tumors that you see in the actual tumors. If you grow these cells in 2D, they lose the pigmentation, which I think tells you that there is really something fundamental about contacts between cells. And I think that justifies doing 3D cultures. We are pretty stringent when it comes to characterization of these tumoroids that we grow. We do both pathology assessment. We have a small pathology group that is reviewing systematically the organoids, making sure the morphology of the organoids is similar to the morphology of the tumor cells, of the tumors in the patients. We're doing this at different passages if we can. When we have xenograft, we're also doing this in the xenograft. But again, this is a systematic assessment that is quite critical because we need to be sure that what we're growing is reflective of a patient's tumor, right? So, I always say there's different ways to think about tumor organoids, right? There's like kind of the precision medicine approach, right, where you're trying to sort of get a model to grow as quickly as possible, so that you can do high throughput testing for different drugs and return the results back to patients. But there's also obviously the idea that you can grow models so that you can do scientific discovery. You can, let's say, I don't know, get a panel of 250 organoids and then test a new compound on this panel. And, it doesn't matter so much to some extent, whether, the tumor that you grow is similar to the patient's tumor. Just, you need diversity. And so that's just two different use. We actually, trying to do both side by side. We're actually not, and not focusing on one, but from the point of view of a precision medicine effort, you really need to make sure that what you're growing is very similar to what you have in the patient. And so this is done also looking at things like some genomic analysis of the tumors at different time points, making sure that, again, there's not too much evolution of the tumor that's occurred. Likewise, as a way to make sure that what you're growing is reflective of a patient's tumor. This is a copy number alteration plot showing that indeed at different passages in the PDTOs here, you don't see much tumor evolution. The next step, I think, is really the critical step, and we've invested with Howard Fine and with the Cancer Center here in a very nice robotic setup to be able to do high content imaging, high throughput robotics sort of testing of hundreds of drugs on these three-dimensional cultures. This is something that now we've optimized really well. ALK at the bottom here is the one who's leading this effort here. So we've actually optimized ability to do high throughput screening, reasonably high throughput screening for hundreds of drugs at the same time and combinations in some cases using this robotic system. So the robot has, pretty much everything. It's like almost like a mini lab, mini robotic lab. It has ability to, it takes 384 plates, for example, seeded with different, organoids within each of the wells in matrigel format. It's able to add the drugs. It's able to sort of, culture the drugs that has an incubator where you can essentially put the plates in and kind of, keep them shaking and at the right temperature for some time, and essentially be able to keep the cells happy. And then the robot takes the cells out, measuring the amount of cell killing using different types of techniques. One of them is really kind of confocal microscopy, looking essentially tracking thousands of organoids at the same time. Each of the plates, there's 384 wells, but each of the wells has hundreds, if not, sometimes thousands of organoids. So we need to be able to keep track of all of these organoids and kind of keep track at multiple time points across multiple days, because we know that response to certain drugs, can take actually several days to occur. And so everything is happening within this robot, which is extremely powerful. These are examples of screens that we did. So this is a bit too small to see here, but we're able to test about 500 different compounds in using this robot. And the compounds on the left side in this plot here are the ones that are most effective at killing this particular organoid in this one patient. We've been using different strategies for this. We started by doing 3 doses, and then for the essentially the compounds that seem to be most effective, at least for one of those doses, we do a full dose response curve. I think it turns out that we are now switching to us doing a full dose response curve for all of the drugs. I think that 3 doses, it was probably not enough to really identify, the right range of doses. And so that's why now we're kind of doing a full DLC for each of the drugs. So a question that I get often is what's in the drug library, right? So obviously we have to be strategic when it comes to what we put in there, right? 500 drugs, you can say, well, that's a lot, but we are putting a lot of well-known cancer drugs in there. We're putting some FDA-approved drugs. We're putting a small number of investigational drugs, for example, drugs where we have clinical trials here at Cornell or maybe at some catering across the street from us. well, we know that we can enroll the patients really, effectively if we find sensitivity to that drug. We're putting a lot of very cheap chemotherapies actually in that panel because, again, I think if we find a drug that's, let's say, a targeted therapy, that's like really expensive, it's like, $50,000 per year. This would be an off-label treatment, right? And it's not likely that insurances could pay for that kind of treatment. So, it may be great that we find a drug that's effective, but if the patient can't afford it, that, is not going to help the patient much, right? So we are putting quite a few traditional chemotherapies, if you want, on that panel, just as a way to maximize the options for patients. I should say that, so this is starting to be returned to patients, as I say, under an investigational protocol. And I think we're seeing some, obviously some uptake from the clinicians, with that data, especially for metastatic patients where the options are very limited. I think this is where this kind of screen, I think, is, becoming very appealing for physicians here. This is just an example. This is an older screen here, but, just to show you the diversity of responses, right? These are 2 patients with colon cancer that was published a couple of years ago now as part of the broader effort to describe the initial setup. As you can see, these 2 patients have some mutations in common. They have APC mutations. They have, other things, and essentially kind of overlapping mutational profile. But we looked at what drugs were most effective within each of these organoids, right? And maybe not to our surprise, the drugs that are effective are actually quite different. Trametinib, MEK inhibitor here, is very good at killing cells from this one patient here. Very poor at killing cells from this other patient here. There's nothing in the mutational content of these 2 tumors that could explain that, at least at the beginning. In fact, really nothing that can explain that, and sort of that's it, which I think really justifies, again, this kind of functional, investigational sort of type of screen. And this is just another plot showing that we can do drug combinations. Some drugs that are not particularly effective as single agent, for example, trametinib in this particular case, celecoxib in this particular case, if you combine it with trametinib, you actually get a very, very strong ability to kill tumor cells from this one patient. And again, this is something that I think has a tremendous amount of promise. I think thinking about how we can use, this kind of modeling most effectively, I think moving towards drug combinations, being able to pick drug combinations that are most effective, I think to me is a great application. Because I always say, I don't know, some of you may have thought about this, but there's really no great way now to pick drug combinations. It's hard to pick individual drugs, that are most effective for each patient. We have an extremely limited sort of toolkit to assess whether 2 drugs are going to work well together just by looking at mutational profiles, even doing, let's say, expression profiles, ability to predict synergy, if synergy is what, we're actually looking for. Sometimes I'm not always sure that's the case. I think, being able to kill different tumor cell population may actually be more important. But, either way, we just don't have a particularly good framework for, identifying good drug combinations. And so I think that, this kind of functional approach, I think, in my view, may actually have a great future, in that sense, to be able to assess drug combinations that are specific for each patient. And the universe of combination is very large, and and then I think we need to have obviously some high throughput way to kind of assess those combinations. One thing that is important is that we just realize that everything so far that I've told you, obviously is a very is based on a very simplified picture of what a tumor is, right? I kept telling you that we shouldn't sort of treat tumors as just like like masses of cells or tumor cells, and I think that needs to apply to obviously these kind of models. Actually, we have a couple of people in our group, including Enrique, who you see on the left side here, who's actually working very hard to build immunocompetent tumor models, especially from TILs. So he's actually managed to be able to optimize the co-culture essentially of T cells coming from tumor-infiltrating lymphocytes, as well as essentially tumor cells from the same tumors. What he does actually to split a tumor chunk into 2. One chunk goes for production of tumor organoids, and another chunk goes towards isolation of TILs. The parameters are a bit different. And then, we expanded TILs using IL-2, and essentially, when we end up having enough TILs, we put everything back together into co-cultures. And we can keep those cells alive for some time. And I think then the question is, what can we do to characterize, these co-cultures? How do we demonstrate that they're kind of functional and that, the TILs, doing what they're supposed to be doing? And so we're doing all kinds of different assays to be able to characterize those TILs, expanded TILs, if you want. I should say we're also extracting T cells from PBMCs as a comparator. And I think from my point of view, I think it's obvious that working from TILs is better because you have T cells that are already in theory reactive. I've already seen the tumor cells, may have been selected, may have become a bit exhausted and therefore not functional anymore. But at least they have the right sort of genetic sort of makeup, if you want, in terms of rearrangement and so on. And I think that it makes sense to work from them. With all that being said, I think the jury is still out, and I think you can see it from this plot here, whether TILs or PBMC-derived T cells, may be better. This is showing 3 different patients here where we did this analysis, and what we did here was to assess co-cultures of tumor cells from the same and T cells from the same patients. Again, T cells either derived from the TILs or from PBMCs. This is looking at different perturbations here, different antibodies that sort of perturb different checkpoints, and looking at what happens in terms of interferon gamma sort of expression, as well as TNF alpha expression. So what you can see here is actually, first of all, most importantly, there's a tremendous amount of diversity between patients, right? The same kind of culture across multiple patients respond to these perturbations in different ways. Sometimes the TILs derived T cells and the PBMC derived T cells kind of agree in terms of how they respond to those perturbations, maybe here and maybe also here, but sometimes they agree less. As I say, the jury is a little still out in terms of whether it's better to work from TILs or from PBMCs. I mean, one other thing that we see is that it's harder to grow a large amount of T cells from TILs. Sometimes you actually are not able to extract a lot of TILs, from the tumors, which is not enough, or they're not easy to extract. So that's something to keep in mind. With that being said, I think we're seeing that, for example, looking at some effective molecules and looking at, for example, things like combinations of treatment with different antibodies targeting different checkpoints, we're actually able to see the T cell respond really nicely, to certain things like combinations of two perturbations. So we see essentially this kind of patient-specific response, essentially, but also showing the feasibility of even looking at combinations and looking at combinations that together are producing an immune response that you don't see with each perturbation alone. And now what we're doing is to essentially set this up as an imaging assay, looking at essentially sort of T cell markers and tumor markers and sort of caspase sort of viability markers or apoptosis markers to look at essentially situations like in this slide where you have a proximity between the tumor cells and the T cells, and essentially a dying cells as a result of this proximity, which allows us to essentially use this kind of system in a high throughput format. And we are still in the early stage of doing this, but I think we're getting promising results. And as you can see at the bottom here, I think some of the data really looks reasonably strong. So this is still sort of work in progress, but I think we're starting to move towards establishing proof of concept that we can use those co-cultures as a way to assess combination of different checkpoint inhibitors in this kind of immunocompetent sort of setting, or combinations of therapies targeting the tumor cells and the immune cells. And we have actually also a proof of concept in this. We have a paper that's about to be submitted on this. In fact, I think we have more data than in my slides, but I think it gives you a sense of what we're trying to achieve here. Just a few more points here. I think that obviously we're kind of interested in the concept of avatars, right? What I've shown you so far is a bit of an avatar. We kind of like to think of it as an avatar. It's like a copy of a patient's disease on which we can test many hypotheses, right? It's a physical avatar if you want. But we've actually become interested in this concept of digital avatars. Can't we think about maybe sort of creating, instead of a physical avatar, creating almost like a digital copy of a patient's tumor, and maybe using that digital copy as a way to test not, let's say, thousands of hypotheses, but maybe like 1 million hypotheses. I think this is the concept of digital twin, if you want, that I think is becoming, more and more accepted. There's actually, a paper on this in cardiovascular disease. In fact, a few years ago, I think we kind of did something that I think maybe we can think of as an avatar of a tumor of a lymphoma. We built an in silico model of a pathway that is absolutely critical to the survival and the function of B-cell lymphomas. We know that because that's a pathway that essentially is responsible for antibody production and maturation and expression and response to stimulations in lymphomas. And we know that again because this pathway is heavily mutated in lymphoma patients. Actually, all these nodes here, this is the pathway here. It has all kinds of different connections. It's actually pretty complex to model, but we know that this pathway is really driving B-cell lymphomas. This has been known for some time now, but I think you can kind of get a sense here of the complexity of the pathway, right? So it's a complex pathway with a lot of different feedback loops, and it's not easy to just look at this pathway and say we should block this particular node or this combination of node and assume that this is going to give rise to shutdown of the pathway and loss of proliferation. So we figured, okay, because this pathway is also critical to the survival of a B-cell lymphoma cell, why not try to model this pathway? And that's what we did. The details don't matter so much, but we provided essentially we created a kinetic model of this pathway, really modeling the feedback loops, the signaling and so on, which involved a lot of finding sort of the right parameters for the different aspects of the pathways. But long story short, we were able to simulate treatment with different combinations, let's say of two different treatments targeting some of the nodes that I showed you earlier, and made some predictions, for example, about which combination would be effective at maximizing synergy and maximizing the killing of these tumor cells, and for example, combinations of treatment that would antagonize each other. So we found, for example, that combination of LIN inhibitor and SYK inhibitors, that was a prediction, was able to achieve maximum synergy in terms of killing. We actually went to multiple cell lines and treated those cell lines with this combination, and we actually saw this predicted synergy in the cell lines, and basically exactly as the model predicted. And the same for antagonism, we actually saw that combination of SYK inhibitor and MOL20 inhibitor, as predicted by the model, would actually be antagonistic, and we actually see that also validated in cell lines. So, this is a very small attempt towards this idea of building almost a virtual model of a disease, kind of an in silico, model of a disease. But I do think that, this is the right approach because I think, once we have an in silico model, we can test, lots and lots of hypotheses, which I think is still a limitation even with tumor organoid models. This is the team of people who are part of the organoid team here, and there's just amazing people doing incredible work. Laura Martin, who's actually left recently, but she really led this team and has done a great job. Enrique is the one who's in charge of the immunocompetent model building. Flor is doing CRISPR screening in organoid models and is getting some really cool results. And Elke is leading the automation platform. So I think that's actually it. That's what I wanted to discuss today. Hopefully we have a little bit of time for question, and just wanted to thank you for your attention, and hope that we can have a discussion.
Thank you, Olivier. This was a fantastic talk. So for questions, yes, you can raise your hand. You can put it in the chat, whatever works. Tony, do you want to get started?
Sure, that was really wonderful. I mean, really, it's great to see. I think it's a really great, very systematic approach you've taken. One thing you didn't get into is like I forget the part when you were talking about. I think it was in context of what Howie Fine was doing, but it could be something else, where you talk about returning results to patients. And I know for many of us, that becomes a major regulatory barrier. So I'm wondering, in order to return results to patients, did you require doing this under sort of a CLIA format? Did you get an IDE from the FDA, or what did your IRB require for you to return this information to patients?
Yeah, so this is done in the context of a research protocol for advanced cancer patients. Essentially, we were lucky to have the IRB agree to using all kinds of different genomic techniques and non-genomic techniques and essentially bring the results of these techniques to case conferences. So what we're doing essentially is we are allowed by the IRB to discuss the results of these analysis in the context of case conferences. We're not allowed to put those results into the EHR, but we're allowed for advanced metastatic patients, essentially patients whose options are extremely limited, to have a discussion about the results in the context of case conferences. So that's how we, at the moment, are returning results back to, let's say, back to physicians. I think that obviously we're doing it. So we have a long history of building CLIA assays in the clinical genomic setting. We're using some of that knowledge and experience to make sure that essentially we're kind of putting ourselves in CLIA sort of type of settings for the entire process. We're working with pathology very closely on this. That these are the limitations. I would say the return of results is not happening systematically. It's happening only for a subset of patients who fit all these different criteria, especially the advanced metastatic stage. And again, with some limitations in terms of eventually, what can go into the EHR. So that's basically what the situation is.
Thanks.
Thank you. Ines, go ahead.
Hello, thank you for the interesting presentation. I was wondering, you mentioned that you have like biobank of 250 of the patient-derived organoids from 250 patients. I was wondering, were these organoids developed from the post-surgery material, or were they developed from the core biopsy? And if so, like, what is the efficiency of development of the organoids? Because can you get the organoids from every patient?
A great question, right? So I'm sure that many on this call probably have at least some ways to answer this, but my answer is like this. So we're trying every time we can get fresh tissue, and every time that essentially we have let's say an agreement with a treating clinician, or let's say there is a patient who we think would sort of fit some of the criteria that I discussed, we try to derive an organoid. We know by now, having done this many times, that it's easier in some situations. It won't surprise anybody on this call that it's much easier to derive a colon cancer organoid compared to pretty much everything else, because that's where partially it's we're still sort of leveraging the Hans Cleaver sort of media, and that media was optimized for colon organoids, and so it works really well in that setting. We are optimizing the media or the culture media in different and other settings to try to maximize our results. We have a big breast cancer project, for example, and I'm sure likewise it won't surprise some of you guys, but our success rate in breast cancer is much more limited. Our success rate in prostate cancer is extremely limited. We've actually tried hundreds of times, and we're able to maybe we have let's say 5 organoids in total. So really very limited. I mean, those 5 organoids are very precious resources, but they are not. They're not. It's very hard to grow organoids. I think general rules is that the more aggressive the tumor, the easier it is to develop an organoid. Another general rule is that if you have a surgical sample with a lot of tumor cells and with a lot of tumor mass, essentially kind of gives you more shot on goals, if you want, and therefore you're more likely to eventually get an organoid to grow. There's a substantial fraction of these organoids here that are coming from core biopsies from metastatic tumors, for example. But at the end of the day, it's it is easier from larger tumors. There's no doubt about it. And so these are some of the parameters, right? The larger tumors give better results, and some tumor types also give better results.
May I have the next question about
course,
this?
yeah.
Do you also consider, or is there some possibility to get also the biopsy of the resection line to have also not only the tumor-derived organoid, but also kind of like normal cell-derived organoid that you can compare then for the cytotoxicity of the drugs?
Yeah, well, another great question. So that's something we're very interested in doing moving forward as part of a screen. We're doing that already. We have a small number of normal organoids that we try to add to some of the screens so that we can test toxicity of certain types of combinations, for example. We haven't systematically been trying to grow normal organoids from those tumors. And I think to some extent, I don't think, you know, there's a situation where there's I think a major need for the normal organoids to be matched to the patients. I mean, I think you can probably, you know, I can find lots of different, you know, reasons why, you know, what I said is probably not particularly sort of, you know, accurate. But I think that, you know, there's been no assumption is that there's more similarity between individuals in terms of normal tissue. And therefore, as long as you have a panel of, let's say, 5 normal, let's say, you know, lung organoids, you know, you should be able to assess safety reasonably well, that's including for combinations. I mean, I think it's a hypothesis. I think we'll see if, you know, that turns out to be true. But I think that's been our sort of that's been our assumption. But yeah, to answer your question broadly, you know, we have derived some normal organoids in some cases, but we don't do it systematically.
Thank you.
Let me ask you a quick question. Have you tried to look at the architecture with the same approach you do for tumor, also for the organoids, and maybe try to match it?
Yeah, we're doing that a little bit. You know, we're doing both imaging mass cytometry on organoids as well as you know, single cell RNA-seq analysis. I think that's the early days. I think we need to do that more systematically because I think, you know, I think to what you're getting at is that, you know, there's even like if you only focus on the tumor cells, right? There's a ton of plasticity within tumors, and we know that, you know, that's the case, right? So one key question is whether that plasticity is preserved in these cultures. You know, we're making some assumptions that's the case, but honestly, I mean, I think it's a pretty, you know, critical thing to assess, right? And so I think that a kind of multiplex imaging of some sort, you know, may actually be a good way to assess you know, this kind of plasticity, or even like, for example, in the core cultures, I think a question that comes up very often is, you know, are we kind of, you know, able to, let's say, infiltrate, you know, the tumor little chunks, you know, with T cells, you know, do the T cells go inside the tumor? Likewise, it's hard to tell without measuring those things, and maybe likewise, you know, the kind of a multiplex single cell imaging may actually be very helpful to establish this.
Thank you. Thank you very much. So we're slightly over time. So I think we're going to let all of you go, but I want to thank you, Olivier, again, to be here with us today. It was fantastic to hear about your work. And I guess we'll see all of you in a month, and yeah, hopefully at the AACR.
Great. Thanks for having me again. Take care, guys.
Bye. Bye, everyone.
Take care. Bye-bye. Bye.